Thank you, Kara. Perfect pronunciation, I have to say. Let's see. I need something to move the slides. There it is. And I need some pointer also. Is it in here also? Yeah. Okay, good. So I think we ended already the talk. So can we back up from the beginning, please? Since apparently, as you know, Sweden is far away and my title didn't make it here for some reason. So I had the chance actually to modify the title during the flight here, which is a long flight. So I called it Targeted Molecular Diagnosis of ME CFS, the devil is in the details. And I will try to explain how I'm thinking about this. And of course, it's a bit accurate to use Tasmanian devil when you're so close to Tasmania here. So we will talk a bit about what we're doing in Uppsala. We will also talk about the collaboration we have. And I think also I want to address that Open Medicine Foundation, Invest in Me, also Solve ME has been very much helping us to actually get collaborative work going, both within Europe and also now with the United States, and hopefully also Australia in some time. But let's go to Uppsala first. Uppsala is just on the other side of the globe, so it's a faraway flight. Alan also already told you that we, on the other side of the world, have a pretty cold weather. So this is last week in Uppsala, no, outside Uppsala. And it's so cold, so the mooses aren't even there anymore. And you can barely see pieces of reindeer sticking up from the snow, so deep snow. Terrible. So it's very nice to be here, and I'm very sorry that I flew in this morning and I will leave in a couple of days. I would love to stay here for a bit more of time, so we had the snow melting away from our countryside. I don't know what you know about Sweden, but what's the most famous thing in Sweden based on Australian people? ABBA. Yes. How did I know? They came here in 1977 on a world tour. Apparently that was set some fingerprints or blueprints of Swedish characters in Australia. I just learned that ABBA will promised not to reoccur again, but they will come in a virtual format 2019 to Australia, actually. So you can go to, I guess, a cinema theater somewhere and see them almost live, virtual reality forms, like avatars, apparently. But as a scientist, and from Uppsala, I would hope that you actually knew something more about Sweden. So I will give you a bit of history about how Sweden is actually related to Australia also in the end. So this is Carl von Linné, a professor at Uppsala University. He was a medical doctor, but he was not doing too well in that field, so he started looking into botany instead and collected plants all over the world, or actually sent his students to collect them. But he gave the names, and all the Latin names that are used for plants and insects, etc., and some animals, are based on his Systema Naturica, which was the Latin book that he wrote. He sent out students, 17 students around the world, and actually some of them reached Australia, as you can see on this map. Many of them died on different places in the world, but the ones who came to Australia actually did pretty well. So they came with Captain Cook. The first one was Daniel Solander. And Daniel Solander, he came to Sydney and to the Botany Bay, and actually that's why it's called Botany Bay outside Sydney, and collected a lot of material. And later on, another student of Linné came called Anders Sparman, as you see. But you see, they've been almost everywhere, even to Canada for some reason. I don't know why. Yeah, exactly. Daniel Solander, he was a pretty happy guy, actually. This is how he looked. He wasn't happy that for so long time, though. He died at the age of 49 or 50. The thing is, we went to these excursions around the world, did pretty well on the ship, apparently. Came across a lot of different small species that he brought. And he was also taking party with a lot of other famous scientists when he was traveling with Captain Cook. So this is supposed to be Australia. I don't know why they actually carried a statue with them when they were getting down to Australia. I'm not sure if that's an artist's decoration only. However, this was also made notice of in Australia, that Daniel Solander came to Australia and found a lot of plants and also helped naming some of them with the ship Endeavour, as you see in the bottom stamp here. So that's actually a Swedish-Australian stamp collection. You can start your collection there. It's supposed to work even in Sweden to send letters with these stamps. However, Daniel Solander, on his way back to Scandinavia and to Sweden, he stopped by in England, and he never came back to Uppsala. And that made his teacher and economic supporter, I would say, a bit upset. So he says, Carl von Linné says about Solander in Swedish, in Old Swedish, den otacksamme Solander giver mig en enda ört eller insekt av det han samlat i insulan Australia, Nova. That means the ungrateful bastard Solander did not give me any of those plants or insects that he actually collected in Australia. He came to London, and he actually became the librarian of the British Museum in the end. And he died in London in the end. A lot of Linnaeus collections also ended up in London later on, because his widow sold most of it to a British collector, actually. But it's preserved in the Linnaeus Society, which is very nice. Okay, over to another exploration then. So what are we doing in ME CFS from our perspective? And I moved around in my slides a bit, so now I'm going to do a bit of proteomics, a bit of metabolomics, and I actually moved some things in here just to tie together with things we have heard earlier this morning. So our focus is a lot on the neuroimmune relative interaction when it comes to ME/CFS and other neurological disorders that we are working with. So just as a recapitulation for you guys, that the relation between the immune system and the central nervous system is very important, and it's also controlling a lot of the functionalities we have in the human body, which is very good in healthy state, but comes quite complicated and could be a disaster in disease states. So we have the interaction with pathological immune interaction when it comes to breakdown in neurodegenerative diseases. Multiple sclerosis is one typical example where we have an autoimmune reactivity against the myelin sheaths, and that in the end kills the neurons. We have a control of the central nervous system by transports or trafficking of immune cell-like structures like microglia. We have immune CNS interactions when it comes to homeostasis. We have the protective immune system when it comes to injury and disease, also in the central nervous system, although most of the time the body tries to control the immune reaction when it comes to regions of central nervous system. So it's often called immune privilege sites, just to address that immune reaction or large immune reactions is not what we want in the central nervous system. And then when it comes to other peripheral organs, we have reactions and maturation processes, and also, as we will come to a bit later, the gut-brain axis, where we have effects of the central nervous system on the gut, and we also have the gut affecting the central nervous system, and also talking about microbiome in the end here. So how did I end up in ME/CFS? Well, very much by coincidence. We've done a lot of studies on central nervous system based on cerebrospinal fluid detection, and we were going to do a collaborative work with colleagues in the United States, where we looked at post-Lyme disease patients or post-treatment Lyme disease patients, or neuroborreliosis patients. And then at that time, we were going to do profiling of these patients. Colleagues came with the first chronic fatigue patients that I've seen. So we used cerebrospinal fluid from both these cohorts, and to make it very short, we compared the patterns on deep proteomic profiling, and we found that there is a lot of similarities between healthy individuals, patients with chronic fatigue syndrome or ME, and these post-treatment Lyme disease patients. But you can also see that there is distinct differences. So we have specific patterns that is unique for the chronic fatigue group. We have specific patterns that are unique for the post-treatment Lyme disease patients, and we also have, of course, in our healthy control specific markers there. But in general, the overlap is very big, as you see here. So if we conclude that study, what we could tell is that there is actually, although low grade, but a neuroinflammatory reaction going, and we have specific protein markers that tells us that when we monitor it in central nervous system by cerebrospinal fluid analysis. We also have signs of increased synaptic plasticity and neuromigration activity, and that is actually not a good thing in this case. It's also the same phenomena we see in other neurodegenerative processes like Parkinson and Alzheimer's disease. And we also see that the ME/CFS patients have definitely a problem with some of their axonal regeneration and repair system. So that is something that could lead to long-term illness and chronic illness in the end. When it comes to the metabolome, we already heard about metabolome today quite a lot, and the complexity of the metabolome is enormous. It is representing the physiological endpoint, and it's, as Alain said also, it's a genetic predisposition and environmental influence in combination. So that is what makes up our phenotype normally and also in disease states. So what makes it so complex? Well, there is a lot of things that we have to take into account. First of all, if we look into the metabolome compared with the proteome and with the genome, we know that the genome is fairly simple in that sense that we have 4 bases that we are building a genome on, although the complexity of a genome can be quite a lot if it comes into epigenetics also. When it comes to the proteome and the proteins built, it's about 20 amino acids. But when it comes to the metabolome, we have 2 to 10 to the 5 different chemicals in there. So thousands and thousands of chemicals that can be affected. And when it comes to the pyramid of life, as you see here in the lower part of the picture, you see that in mammalians, we're actually not so complex when it comes to the metabolome. We have 20,000 different chemicals approximately in our bloodstream that we can measure. When it comes to microbes, the metabolome gets much more complex, so about 60,000 different chemical substances. And when it comes to the plants, there are over 200,000 chemicals that have been detected now and mapped for the metabolome. So in that sense, the mammalians are less complex, of course. Then we should remember that quite a few of the things that we find in mammalian samples can also be derived from food intake of different plants or by the microbes production in our intestinal tract, for instance. So when it comes to a metabolome study then, or a proteome study, we can make this a quite complex history because the technology today has developed so much, and I think Ron spoke about this in the morning, which I unfortunately missed. But the complexity of the big data and handling big data sets is something that we face today. So I just made this very small calculation to show how complex it can become. So let's say we select a patient group of 20, very small pilot study. We have half women, half men. We take samples from them, maybe 10 different tissue samples or body fluids or fecal samples. We maybe collect a few samples per day, and we do it over time, so let's say 10 different time points, just to try to map the omics in these individuals. So that only makes up 10,000 samples, just that little 20 individual study. And let's say we make 2,000 proteins per sample in general in these sets. and maybe 2,500 metabolites per sample. So then we come up with about 20 million protein signals that we have to take care of, and maybe 25,000 metabolomic signals that we have to take into account when it comes to doing data analysis in the end. So of course, data handling and big data handling is something that is crucial in the field. When it comes to the metabolomic then, we have two strategies in Uppsala. We do quite a lot of targeted metabolomics, where we actually are, from the beginning, selecting a pathway or a family of molecules or a known set of just a few molecules of interest. So we actually know what we're looking for, and then we can use standard technologies. We can do quantitative measurements, and we can know for a selective set of markers what we are doing. So the technology is normally liquid chromatography, tandem mass spectrometry, high resolution, or as you will see, ultra performance convergence chromatography and tandem mass spectrometry. So we do selective set of markers. We do standardized techniques for measuring quantitative. We correlate the findings with different locations in the body or different biological systems. It can be tissue type or cell type. And then we do variability tests and look for differences in controls in patients, for instance. So the usefulness of this is quite high, because we can set an absolute quantitative level. So that data is possible to translate to other research groups. We can send out nanogram per ml, or we can send out femtomolar concentration, for instance, of each individual substance, and that makes it translational in another way than the untargeted metabolomic profiles are doing. So in this untargeted approach, we don't know what we're looking for, so we just try to screen for everything. And we can do that in all different sets of samples by tandem mass spectrometry connected with liquid chromatography or other methods in advance, or using NMR, as we also heard earlier this morning. And then we just do advanced pattern recognition initially, align the data sets, and try to figure out if there are any differences, what are those differences related to. So we in the later stage of the analysis find out, hopefully, what it can be. Sometimes we can't even identify what is the difference. We can see that there is a difference in the pattern, but we're not able to identify what the difference is due to. Very useful for exploratory work and for finding new targets, but also a problem is that quite often we don't get absolute quantitation, so we can't really get the translational part. We get relative quantitative data, which can be a problem. And it also, some of these data sets that are out in literature based on untargeted metabolomics is also showing quite a big variability and not reproducibility when it comes to different groups around the world. So a problem there. So just an example of a targeted strategy then, and this is something we've been working with for quite a while with different disorders, but I thought of ME/CFS as one target for targeted steroidomics then, when we look at steroid and steroid hormones. And steroid hormones are very important for a lot of things. There are neurosteroids that control synaptic plasticity and synaptic regulation. But steroids can also be very much involved in the regulation of the immune system. So this busy cartoon here shows that when we have a steroid binding to a specific receptor, this receptor can actually be internalized into the cell nuclei and also control a lot of the mechanism in the transcription inside the cell and nuclei. And also affecting, for instance, inflammatory stimuli as you recognize here, interleukin-1 beta, TNF-alpha, etc., and actually block or stimulate depending on what steroid is binding into the receptors and internalized into the nuclei. So that will affect gene level and also affect the stimuli or result of the stimuli we have on the cell. The biosynthesis of steroid hormones is pretty complex. This is just actually one part of the steroid hormone profile. If we look at, for instance, estrogens here, we can actually fold out a tree of 16 other metabolites just from estrogen in that region. So the pattern gets extremely complex. But when you look at the pattern like this, you can see that we have specific enzymatic pathways that are regulated, and we can also know that many diseases when it comes to steroid hormone dysfunction is related to blockages or less efficient enzymes in different levels, and that will affect a lot the end product of the steroid hormones. So what we did in our set, we developed a new method, you could say, for detecting all of these steroid hormones at one time point. A lot of the earlier techniques only could measure 1 or 2 at a time, but we can monitor the whole pathway in this assay now, including 25 different steroids at the same time. And it's based on a technique called supercritical fluid chromatography, or ultra-performance convergence chromatography is another name, and then connected to tandem mass spectrometry. And the method is published now, so you can have a look if you're interested. It's a quite simple technique. You take your body fluid, plasma for instance, you do a liquid extraction of the steroid markers, and then you derivatize them, and then you run high resolution separation and tandem mass spectrometry for the detection. And we get down to picogram per ml levels in most of the steroids here, low level of those. So in the study we have done, so far not published, and this is supported by Open Medicine Foundation and also by the Swedish patient organization, RME. We took Swedish patients, 24 well-characterized patients, and we used 24 healthy controls, sex and age matched, collected at the same time, handled at the same time, stored, following very strict protocols. So trying to avoid variability in the sample set as much as possible, and also including quality control and quality analysis samples in the set. So what we found is that we have a fairly high variability of these steroids. This is just a subset of the data, but you can see that we have our control and the ME CFS group next to each other. You see that there is a large overlap, but also a large spread and some outliers in the data set. And the only steroid that popped out as significantly different is the pregnenolone here. And if we remember how the pathway is, we see that the pregnenolone is a key metabolite of the steroid hormone protein profile, or steroid hormone profile. We did not find a difference in cholesterol in our study. We have very strict diet control of the patient we collected, so that could be a reason why they didn't discriminate so much between the two, patient and controls, I don't know. But you see this one came out, and I also made blue marks here, and the blue marks shows that enzymatic activity for producing pregnenolone, for instance, is in the mitochondrial membrane, and also for other metabolites here in the steroid profile, the enzyme is actually sitting in the mitochondrial membrane. And although these did not pop out significant different in our groups, they were altered, not significantly, but tendency to be altered. So what we conclude is that pregnenolone is, in our data set, differently regulated in the ME CFS patients versus controls. It's a neurosteroid, so it's highly produced in the central nervous system. It is a synaptic functioning regulator, it is neuroprotective, and it enhances myelinization. And it improves cognitive and memory function, so a reduction of pregnenolone could definitely give some of the symptoms we see in the ME CFS patients. And it's produced in the mitochondria, why we believe now that the mitochondrial dysfunction could be one of the reasons for this finding. And this data set that I will go through now is just came to us and is just now submitted, and it's a collaboration with Simon Cardent Group at QIB, the Quadrum Institute of Biology in Norwich, UK, and this study has been supported by the Invest ME. And I took it because I saw that there were discussions about severe ill ME/CFS patients. And what Simon and his student, Daniel Verpond, have done, they have collected at home patients with severe ME/CFS, mostly bedbound, and also been able to have household controls collected at the same time. And they collected fecal samples and plasma from these patients and controls. And I was involved in the metabolomic profiling of these patient samples then. So the idea and the thought about this study from the beginning is the gut-brain axis relationship that some of you know much about. But in a normal healthy individual, we have a relation and a co-regulation of the central nervous system activity through the HPA axis, for instance, on the steroid hormone level, on the gut function, and also vice versa. A normal functioning gut without leakage, for instance, will also have a positive effect back onto the central nervous system. So if you eat healthy diet, if you have you don't have the problem with probiotics, or you get the probiotics there, you write the good biotics, the good microbiome. They put in this slide, breastfeeding and natural birth, which that is affecting the microbiome. Then you have the healthy brain-gut axis going. When you have stressors, C-section is here, formula feeding, but unhealthy diet, infections, stressors of different kinds, antibiotics could be one. You get the unfunctional gut-brain axis then, which leads to dysfunctional regulation of central nervous system, dysfunctional regulation of the steroid hormone levels, a leaky gut, and a changed and altered microbiome in the end. So bad side and good side in this case. So what was done in the study was that NMR was used for the metabolomic profiling, and they looked at initially at the fecal water. So fecal samples were extracted by a buffer basically, and then the NMR was done for metabolomic profiling. And to the surprise, I would say, not so much came out of this. What came out as positive, significantly difference between the severe ME patients and their household controls was a slight change in the glycocolate and also in a butyrate metabolite. So you see there's a big variability and large overlaps in all the metabolites that were screened for here. But if you take them all into account and you do a principal component analysis, you can actually pull out that the severe ME patients is grouping here, and you have the household controls in the green here. I think the laser is dying on me here. Hope you can see the green color at least. So this is based on 45 different metabolites that were made absolute quantitation on. And with the principal component, you can see these differences pop out. But the differences are not very, very large, I have to say. Also, based on earlier studies and also on the findings, we also did a MS-based analysis, mass spec-based analysis, and focused then on fecal bile acids. And as you see, not much turned out, but there are some small differences in the bile acid profiles when it comes to the red is severe ME patients, and the green is the household controls then. But as you see, also a large variability and large overlap between the different groups. Then serum samples were done, and also with NMR for looking through 53 quantitatively identified and quantified molecules in circulation then, and nothing really came out there. It's a large overlap, big variability. Nothing came out between the two control and patient groups, unfortunately. So summary for what I've published or what I'm talking about today is that what we have found is that we have a significant and also repeated finding of low-grade neuroinflammatory markers in cerebrospinal fluid from ME/CFS patients. We just followed up with a targeted approach that I will present next month when the data set is fully available. We have done a panel of 1,300 neuroinflammatory markers and other markers with a targeted analysis. So, and also compared with multiple sclerosis patients, and it looks very, very interesting. That's as much as I can tell right now. But next month, if the data will be available. As you saw, we have this disturbed steroidogenesis found in the ME/CFS patients in circulation, possibly related to dysfunctional mitochondria. I have not mentioned today the data set that we have done in collaboration with Carmen Scheibenbogen in Berlin. We have looked at autoantibodies in circulation from ME patients, and we could, in our data sets from 2 Swedish cohorts, repeat the data that Carmen has published, where we found in a set of patients significantly elevated level of autoantibodies against adrenergic and muscarinic receptors. We did not find anything in cerebrospinal fluid, though, which is sort of a positive signal. If there is a problem with these autoantibodies, we don't need to remove them from the central nervous system, at least. They are in the periphery. And then finally, the study in collaboration with the group in Norwich, then, that these small metabolic or metabolomic alteration was found in the gut of the severe ME patients compared with their household controls. But the differences are not remarkable, I would say, in our hands. So the devil is in the details. And in the end of the day, I think we have to consider the quality of the sample and methodology that we are using. So we can have a method that is, well, if it's really poor, we are looking at something that is not really hitting the target. It's both unreliable and invalid. We can have a method that is fairly unreliable in reproducibility, but still centering around the target, so it could still be valid. We could have a method that is super good on reproducibility, but totally off target. Or hopefully we get a method that is both reliable and valid and actually hit the target. So I think still when it comes to ME/CFS, we are sometimes seeing this, we are sometimes seeing this. In some cases, we are actually hitting the target with a very valid method. So combination of the data sets that are now gathered around the world, I think will lead us in the right direction. But I want to conclude with that we still have to keep on exploring. We have to come across things that are reproducible and that actually tells us what actually is going on. And I think the investments and also the encouragements that we get from both patients and their relatives, healthy controls that voluntarily give samples in these studies are fantastic. The support that we get from Open Medicine Foundation, well, in Sweden we get from the Swedish patient organization RME, Invest in Me in UK, and then clinical collaborators that we are working with in setting the sample set and also collecting material. So I want to express a warm thank you to all of them and also to you for your attention. Tack så mycket.
Have we got any questions? Oh, here we go.
Jonas, you made the really important point that in order for us to compare metabolomics work from one group to another, we need to do it quantitatively.
Yeah.
And so I wondered, to what extent is it feasible to quantify a much larger number of the metabolites that we run through in a metabolomic study?
Yeah. I mean, it could be done, but the problem, as you know, is that to get internal absolute stable isotope labeled quantitatively secured internal standards for everything is going to be very expensive. one can get around it by having group internal standards that group per family of molecules, but it's not optimal. So if you really want to do it quantitatively, we have to have a stable isotope, or if it's mass spec, stable isotope labeled standard, which is a limitation, definitely.
And so to what extent is that susceptible to a higher throughput, lower cost? Is it what's actually limiting in producing those standards?
I think in principle they are not that difficult to produce. It's just that the demand for certain metabolites, for instance, small molecules, has not been high enough. So the companies that actually are doing this for commercial reasons, they don't have them in their catalog, first of all. And if you want to order it, you have to do special orders, and then it will be very expensive.
Right. So it seems like metabolomics is almost an industry already. And so shouldn't a collection of such standards be something that has a big enough market? Absolutely.
I think so too. We buy quite a few of our standards we buy from Canada, actually. They are very good on doing some of these labelings that, and they have some special companies that are doing for lipids, for instance, and also steroids. But I think, I mean, one can do labeling yourself also to some extent, collaborating with a good organic chemist, for instance. But you have always the problem with getting an absolute quantitative measure of that specific standard, and you also have to make sure that the purity is at least 99%. So that is still a problem.
Yeah, thank you for your comment on pregnenolone. I've noticed in patients over the years low pregnenolone, and we have replaced that. And in some patients that has raised their whole profile and has helped their brain. Others hasn't. But we can no longer test it in Australia. It's been removed from our.
Okay.
There was a laboratory we used to use in Melbourne, but I've got no access to measure it anymore.
We get sample shipped from all over the world, so just send me an email so we can set that up. I think how many of the patients approximately was it who were responders of the supplement?
Most responded. I tried to, if I found an isolated low DHEA or cortisol, I'd go around the clock on the edge of the outflow of adrenals and measure what I could and come back to my other hormone. I didn't know which enzymes were affected. I don't know if it was the side chain cutting from cholesterol, the side chain cutter to form pregnenolone that was affecting, or if it was those within, because I can't measure pregnenolone. But previously when I could, if I replaced pregnenolone, I got some good results.
Interesting.
Can I just sneak in again and ask another question? Just on the fact that you use the controls that were within the same household, I know if you look at my diet and my husband's diet, they're going to be two very different things. Did you give them a specific diet to take, or was it just sort of what they ate naturally?
Actually, for the collections, which Daniel was spending a lot of efforts on, they tried to make sure that they were on a fairly strict diet, but you know, one thing is wishing for it and doing it. But I think actually what we see is that the differences, I didn't tell anything about the bacterial strains and so on that they have also monitored in all these samples, but the variability is very small actually. So I think they were doing quite well. Only confounding factor, of course, could be that when you have a household control, it's someone, in these cases, they were related because it was quite often mother and daughter. There is an age difference. Okay, there's no gender difference, but that's, yeah, so that can be one thing to look at. But we were surprised, and I think, many of the others that were involved in this study also were surprised that the variability is so small when you go for household controls. Thank you.
Thank you very much, Dr. Bergquist. So thank you to all three of our speakers again from this session. It's been very informative, and I've still got a lot of questions. So we'll take a break now for tea and coffee before the final session of the day. So thank you.